Welcome to the Myotonic Dystrophy Foundation's Meet the Drug Developer session for today, Friday, August 4th. A little bit about the foundation. We envision a world with treatments and a cure for myotonic dystrophy. Our mission is community, care, and a cure. We support and connect the myotonic dystrophy community. We provide resources and advocate for care. We accelerate research towards treatments and a cure. Just a refresher about the resources and support available from MDF. Our toolkits and publications, including our clinical care recommendations, are available at myotonic.org/toolkits-publications. Our support programs, including information about our support groups, can be found at myotonic.org/find-support. Our calendar of events at myotonic.org/calendar/month. And our digital academy, which is host to all of the presentations, including conference presentations and today's program in the next week or so. They're all hosted on our digital academy at myotonic.org/digital-academy. So as a reminder, this presentation is being recorded, and so it will be posted on the Digital Academy within about a week or so. We would like to make sure you are all aware of our conference in Washington, D.C. this year. We are only about 1 month away. It starts on September 7th with the MDA Advocacy Day on Capitol Hill. We are grateful to Avidity Biosciences for being the lead sponsor of our conference this year. The general program will begin on September 8th, and we hope that all of you will be there. You can see more information about the program and the speakers and the mentalist and magician who will be there at myotonic.org/conference. Okay, you are here to meet some of Avidity Biosciences' phenomenal team. On deck today, we have Sarah Boyce, Avidity's CEO and President. She joined Avidity in October 2019 and serves as a member of the board of directors. She brings to Avidity extensive operational, commercial, and leadership experience in the life sciences industry and has built global organizations, bringing a number of innovative therapies, including Tegsedi, Waylivra, Soliris, Gleevec, and Tasigna to patients. She most recently served as president and a member of the board of directors of Axia Therapeutics, where she led the commercialization of the company's rare disease products. Prior to joining Axia in April 2018, Ms. Boyce held multiple executive-level roles at leading pharma and biotech companies, including Ionis Pharmaceuticals, Forest Labs, Alexion Pharmaceuticals, Novartis Oncology, and Roche. She currently serves as a member of the board of directors at OmniAb, Inc., and she received her Bachelor's of Science degree in microbiology from the University of Manchester in England. Also on deck today is Dr. Steve Hughes, Avidity's Chief Medical Officer. He joined Avidity in February 2022 with over 20 years of experience building and leading clinical development and medical affairs teams. He's contributed to over 50 clinical trials for more than 25 drugs across multiple therapeutic areas, including cardiovascular, neurology, and several rare diseases. Dr. Hughes joins Avidity from Arcturus Therapeutics, where he continues to be a strategic clinical advisor. Prior to that, he held the position of Chief Medical Officer, where he provided leadership and direction to clinical operations, clinical development, clinical sciences, data management, biometrics, and drug safety. Dr. Hughes' previous positions include CMO at Organovo, Chief Clinical Development Officer at Ionis, and positions at Biogen, CSL Behring, and Sanofi. Dr. Hughes is board certified in pharmaceutical medicine and received his medical degree from Imperial College London. He also has a master's in business administration from Imperial College Business School. On Avidity's team with us today is Dr. Lee Tai, Avidity's Executive Director of Clinical Development. She joined Avidity in September of 2020 and has been contributing to the development of AOC 1001. She has worked in the life sciences industry for more than 10 years as a medical monitor and clinical developer on phase 1 to phase 3 interventional trials in the areas of cardiovascular outcome, metabolic, and neuromuscular diseases. This includes supporting the development and approval of Tegsedi and the treatment of transthyretin polyneuropathy, as well as the development of its second-generation compound. Prior to that, Dr. Tai focused on basic science research in the field of arteriosclerosis and took care of patients as an attending physician at the San Diego VA hospital. Dr. Tai attended Northwestern University in the Medical Scientist Training Program for her MD PhD degrees. With us today is also Kristi Clark, Avidity's Vice President of Clinical Operations. As a Senior Strategic Leader for Clinical Operations, Kristi develops and executes global clinical operations strategy across all development programs and ensures clinical study conduct and integrity are maintained to highest standards and best practices. Kristi has over 30 years of experience in global clinical operations, including greenfielding an office in Czech Republic. Kristi has worked in the rare disease space since 2005 and co-founded Agility Clinical Inc. in 2012, a rare disease-focused clinical research organization. Kristi has a son with a rare disorder and is passionate about ensuring that the patient is a true partner throughout the clinical trial process and educating the patient on how their voice is heard during participation in natural history, registries, and clinical trials. And another member of our team today will be hearing from Luis Estevez. He is Avidity's Senior Associate of Patient Advocacy. He joined Avidity in 2020 as a research associate and contributed to research efforts across the DM1, Duchenne, and FSHD programs for over 3 years before transitioning to the patient advocacy team in April of this year. Witnessing Avidity's patient-centric approach to drug development firsthand inspired him to take on a new role in patient advocacy and continue working to amplify the patient voice as Avidity aims to bring meaningful treatments to the community. Luis attended the University of San Diego, where he received his bachelor's degree in biochemistry. Okay, thank you to everyone who submitted questions in advance when you registered for today's program. Our participants today and our speakers have done what they can to incorporate answers to those questions into the presentation. If you have questions, you may ask them live. You can type them right in. You just go to the questions tab on the control panel, type your question, and click send. If you are on a smart device or smartphone and you see a question mark icon at the top of the screen, you can type your question there and click send. If today's program speaks to you, please consider showing your appreciation to MDF. We do work hard to make sure that we can connect our community to people who are developing drugs and to clinical experts. Together, we are going to change the future of this disease. Myotonic.org/donate. And you are all here to see the phenomenal Avidity Biosciences team. So thank you for joining us. And without further ado, I would like to welcome them to the stage.
Thank you so much, Tanya, for inviting us to be with you today. It's always such an honor to partner with the Myotonic Dystrophy Foundation and also to have the opportunity to speak to the larger myotonic dystrophy community. If we can go to the next slide, please. During the course of this presentation, we will be making forward-looking statements, and that full forward-looking statements is available on our website. We can go to the next slide, please. A little bit about Avidity and what we're trying to do. We are a team who's deeply committed to improving lives of people impacted by many rare diseases, including myotonic dystrophy. And the way that we're trying to do that is through the development of new drugs. such as AOC 1001. Avidity was founded over 10 years ago. We are a publicly traded company. Our ticker, our NASDAQ ticker symbol is RNA. Our home base is beautiful, sunny San Diego. We are a platform company, and we're delivering a new class of RNA therapeutics using our technology called antibody oligonucleotide conjugates, or AOCs. We have 3 programs in the clinic today, and we are engaged either in the clinic or in research in many therapeutic areas, including in skeletal muscle, immunology, and cardiology. If we can go to the next slide, please. Now, just to focus in on AOC 1001 and some of the milestones of what's been happening. So in August of 2021, our IND was accepted. That's the point where you have a go to be able to start your clinical trials. And the first participant in the MARINA study was administered drug in October of 2021. In August, we also were able to, August of 2022, we were able to enroll participants in the MARINA open label extension study. So having completed MARINA, participants were able to go into what's called an open label extension study where they can continue to receive drug. And then we've had a series of announcements of the incredibly exciting data, which Lee and Steve will speak to, around both in December of last year and then also in April of this year. Today, having completed the MARINA study, we are now deep into our preparations around finalizing pivotal study design, which will be a phase 3 study for adults living with myotonic dystrophy, to be able to move the program forward as expeditiously as possible. With regards to today's agenda, which is on the next slide, I am now going to hand over to Lee Tai, who is our Executive Director of Clinical Development and leading the AOC 1001 program. And Lee and Steve will then take you through that. You'll then hear from Kristi and then back to me at the end. And then we would love to make sure we have time for a Q&A as well. So without further ado, Lee, over to you.
Can everyone hear me? Yes?
Yes, thank you.
Okay, great. Thank you. All right. I'd like to spend the next 10 minutes or so to share with you a little bit about our drug AOC 1001, which Avidity developed from lab bench to bedside in our clinical trials. But first, I want to acknowledge the participation of patients like yourself over the last several decades, who allow the physicians and scientists to study them and their diseases, as well as that of their family members. This enables the researchers to build an understanding of what we now know about the DM1 disease, and it's very important and really build a foundation for us to develop a drug for this disease. To support the discussion of our drug, I need to first set the stage and talk about DM1. DM1 is caused, as you know, by a mutation in the gene called DMPK. The mutation causes hundreds and thousands of nucleotide repeats to build up at the end of the DMPK gene. When the mutant DMPK gene is transcribed to RNA, the RNA also contains these repeats, and these repeats are toxic to the cells and impair normal cellular function. On the next slide, the ultimate result of this toxicity is DM1 disease signs and symptoms that can affect multiple organs in the body. In the next slide, a substantial amount of this disease signs and symptoms are in the muscular tissues, including skeletal muscle, smooth and hard muscles, as shown in this slide. These muscular symptoms can present as myotonia, hand weakness and stiffness, pain, GI issues, abnormal heart rhythm and function, and respiratory or breathing problems. This results in a significant decline in quality of life. This is an important point for the drug because we are developing for DM1. The drug is specifically targeted to go to the muscle and heart tissue, and the goal is to reduce the amount of toxic DMPK RNA made by the cell. And we hope that by reducing the toxic DMPK RNA level in the muscle and heart, there will be a decrease in some of these signs and symptoms. Now, in the next slide, I'd like to tell you a little bit about the AOC 1001. First, I will share with you a little bit about our innovative drug technology, which unlocks a new way of delivering drugs directly to the affected cells, like the muscle and heart cells. And this allows us to develop new treatments for diseases with currently limited therapeutic options. We're applying our technology first to develop the AOC 1001 for the DM1 community, which has the potential to address the root cause of the disease. In the next slide, we will talk a little bit more about the AOC. The AOC platform really combines an proven technology of existing drug types that are well understood for each of their specific properties in drug development. For the first part, the antibody, which is a naturally occurring protein made by all of our bodies to help fight infections. And antibodies are commonly used in drug development because they can be made to target almost any protein, including those that are different between one cell type and another. Antibodies are the foundation of over hundreds of proven drugs, many of which are designed to treat chronic conditions. Currently at Avidity, we're using the antibody as a vehicle to deliver our drug to muscles and heart cells. So the next component is the oligo. The oligo is a short strand of DNA or RNA. Oligo can be a powerful drug because they can change gene expression or how the drug make a protein by targeting the RNA that inscribes the protein. There are currently multiple approved oligo drugs, many of which are disease-modifying treatments. At Avidity, we use oligo in our AOC that are specifically created to target the root cause of disease, in this case, DMPK. In the next slide, for AOC 1001, the type of oligo we are using is called small interfering RNA, or siRNA. It works by harnessing a natural biologic process in the cell called RNA interference, which is a mechanism used by our bodies to fight off things like RNA-containing viruses. So what happens is that when siRNA is loaded into a specific protein complex, which is shown here as a Pac-Man, it will chew up the target RNA, in this case, DMPK. So let's go on to the next slide. So this slide really puts it all together to show how AOC 1001 works. The antibody we're using sticks to a certain receptor on the cell surface, and this receptor is called the transferrin receptor 1. When the antibody part of our drug finds and binds the transferrin receptor, this receptor pulls the antibody oligo conjugate into the inside of the cell. And once inside the cell, the oligo is let go, and it can go into the cell, both in the nucleus and the cytosol, and bind to the protein complex and chew up target DMPK RNA. So let's move on to the next slide. So now we're very excited to be able to share with you today information about our clinical development program for AOC 1001, including both the MARINA and MARINA open label extension clinical trial. But first, in the next slide, we just want to really express our gratitude. It's really important to acknowledge the important contribution that patients have helped us to get to where we are today. We have leveraged really key component of our development pathway by leveraging a network of advocates, experts, and knowledge that's been developed over the last few decades, and tools including the Myotonic Dystrophy Clinical Research Network, the NDM1 Natural History Study, and our patient advocacy partner, including MDF and the DM1 community of patients, families, and caregivers. Let's move on to the next slide. So this slide is going to tell you a little bit about the trial design for the MARINA trial. The three main goals of the MARINA study are to evaluate the safety and tolerability of AOC 1001 and to understand how the body is processing and break down the drug, and also to evaluate the ability of AOC 1001 to reduce the DMPK RNA in muscle biopsies. There are two parts to this study. Part A and B. In Part A, participants receive a single intravenous dose of either 1 milligram per kilogram of AOC 1001 or placebo, and were then followed for 6 months. In Part B of the study, participants receive either 3 IV doses of 2 milligram or 4 milligram of the drug or placebo. After treatment and follow-up period, patient could elect to enter into the extension trial called MARINA OLE. Currently, 38 patients have completed the MARINA study, and 37 patients are currently enrolled in the MARINA open label extension. And now we have, continued to collect data across the trials. Now this MARINA OLE trial, and we have over 132 doses of AOC 1001 given, and over 27 patient years of follow-up as a whole. And this trial will continue, and we hope to generate more data over time. In the next slide, so we just want to tell you how the patient input has helped us to overcome key barriers in clinical trial development. And we want to thank the community again for their input in MARINA Trial. Through focus groups, surveys, and meetings with our rare muscular disease patients and caregiver council, we have learned about the barriers that can make it difficult for patients to participate in the clinical trials. So we have created supporting services to help minimize some of these barriers, and this includes finding ways to reduce travel and visit to clinic, and when travel is necessary, to support the patients and caregivers, the travel costs and other associated costs. And also we really seek input from the DM1 community to help us understand key challenges and to improve the overall experience for our trial participants. And next slide, we really want to highlight the importance of the participants of these trials to help us understand how our drug works for the DM1 disease. We really want to thank each of them and their family, and also caretakers, investigators, and their team for their time and commitment and their continued contribution in the MARINA OLE study. Now I'm going to pass it over to Steve to talk about the data.
Thanks to the listeners that are online. I'm really excited to be able to share with you today top-line data from our MARINA study, which is our first study of AOC 1001 in myotonic dystrophy type 1. I'm going to start with a summary of safety results and then look at the effects of AOC 1001 on some of the measures of muscle function that we assessed in the MARINA clinical trial. So if we can move to the next slide, please. Great. So I'm going to start with safety. This slide shows at a very high level the adverse events that were reported by participants in the MARINA study. Overall, the safety and tolerability profile of AOC 1001 has been favorable, which is very encouraging as we plan for the next stage in development of this drug and our next clinical trials. As we know, DM1's a serious disease, and if we look in the table at the top row, we'll see that, as expected for a disease such as this, nearly all participants experienced an adverse event of some kind. Even the adverse events in the placebo arm occurred in around 80% of subjects. Importantly, nearly all of these events were mild or moderate. There were 3 events that were reported as severe, however, and I'll talk to those in just a little bit. The most common adverse events are shown in the text to the right-hand side of the slide, and these are procedural pain and anemia. The procedural pain was primarily related to the muscle biopsies, which were part of the clinical trial. As you can imagine, these aren't a completely pain-free experience, and we're grateful to the participants for allowing themselves to be subjected to this. We had some cases of anemia that were reported as well. These were all mild to moderate. They're transient. The anemia is related to the drug's method of getting into the cells, which, as Lee said earlier, is by binding to the transferrin receptor. This receptor is also used for getting iron into red blood cells. So binding of our drug to this receptor temporarily reduces the availability of iron to the red cells, and this causes the number of red cells to be temporarily reduced a little bit. If we look at the third row down in the table to the left, and apologies for jumping around a little bit, but we've kind of got the data to the left and the explanation to the right, so it's a little bit of a juggle. But if we look at the third row down in the table, we see that 2 participants had adverse events that were classified as serious. One of these was at 2 milligram per kilogram dose, and that event actually was not related to the study. It was a consequence of opiate pain medication that was administered to this participant after a minor surgical procedure. And then the other serious event occurred in the 4 milligram per kilogram dose. This was a very rare type of stroke that was reported by the investigator as related to AOC 1001. This event resulted in FDA placing the study on a partial clinical hold. And if we look at the text on the right-hand side of the slide, we'll see this event's been described as bilateral ischemia in the region of the lateral geniculate nuclei in the thalamus. This is really just medical speak for the type and the location of this rare stroke. So now, finally, just going back to the 3 severe events that I mentioned earlier. 2 of these were actually also the serious events that I just described. So events can be both severe and serious in terms of the way that they're reported in clinical trials. The third severe event was related to a procedure. So all in all, 2 of the severe events were felt to be unrelated to AOC 1001, and the event of a rare stroke was judged by the investigator to be related. Could we move to the next slide, please? So now I'm going to move on to the impact of AOC 1001 on some of the functional measures that we looked at in the MARINA study. And I'll walk through these just over the next couple of slides. Just as a point of orientation, on all of the slides, we're showing the change from baseline in the outcome measure that we're assessing. All of the functional measures we assess are also being followed in the NDM1 natural history study that's being conducted by the DM1 Clinical Research Network, and they've therefore been determined to be important assessments for tracking progression of DM1 over time. So the first assessment that we looked at, certainly that I'm going to describe today, was an assessment of hand function. And we all use our hands to perform many activities that we do on a day-to-day basis. So hand function, as you can imagine, is extremely important, and loss of hand function can be very debilitating. We used video recordings of hand opening time to assess function of the hand in this particular assessment. So it's called video hand opening time, or VHOT for short. And in this assessment, the participants are asked to make a fist as tightly as possible, then to open their hand as quickly as possible, and you'll hear this on a video in just a minute. So the assessment's measuring both the impact of myotonia and the impact of muscle strength on the ability to open the hand quickly and in a normal fashion. So if we just look at the graph on the right-hand side of the slide, this is showing the average results for all of the participants in the study. The grey line is placebo, and the other lines, as we move down the slide, are the 2 milligram per kilogram cohort and the 4 milligram per kilogram cohort. And we can see that certainly in the 4 milligram per kilogram cohort, we're getting good separation from the placebo by about week 6. At day 92, which is month 3, we're seeing some separation at both 2 milligrams per kilogram and 4, and the 4 milligram per kilogram improvements are maintained out through to the end of the study, which is at 6 months. So this is over a 3-month period with no further dosing. We see that at 4 milligrams per kilogram, the treatment effects maintain, whereas at 2 milligrams per kilogram, it goes back towards baseline. If we can move to the next slide. So this is a video of how this test is performed. We're just showing it for one participant from the 4 milligram per kilogram dose. And we're showing the values at the baseline, which is on the left, and at the end of the study, day 183, which is 3 months after the last dose. So a couple of things to focus on. One is the how long it takes on the right-hand side for this participant to open their hand, and we can see how that level of impairment could be very disabling in terms of performing activity. So.
Good, and just stay there.
The two videos are actually run synchronously in terms of from the beginning to the end of the test. So this is showing the ability to open the hand at the end of the study versus prior to receiving any treatment. I think we can all see that on the right-hand side of the slide, the participant is, or at the baseline, the participant is takes a very long time to open their hand, whereas 3 months after 3 doses of study drug, we're seeing very noticeable improvements in the study. And we've seen this in many of the participants that were treated with AOC 1001 saw notable improvements in their hand opening times, as was shown in the graph previously. Could we move to the next slide, please? So the next assessment is a measure of pure muscle strength, it's called quantitative muscle testing, or QMT. And in this assessment, we measured the strength of 3 upper limb and 3 lower limb muscle groups, and we compiled them into an overall score, which we call a composite score. So this is the composite QMT score, and it's based on % of normal muscle strength. So if we, so that you can see to the left of the slide here, these are the muscle groups that we evaluated. So there's hand grip, elbow flexion and extension, knee flexion and extension, and then ankle dorsiflexion, which is ability to raise your foot upwards. The right side shows the improvement, and so we can see here that upward direction shows improvement. The grey is placebo again. We can see that at about 3 months, both 2 and 4 milligrams per kilogram are separating from placebo. This continues to improve over the time period out to day 183 for the 4 milligram per kilogram cohort, and decreases a little bit over that time for the placebo. But I think it's clear to see that we are seeing directional improvements at both doses on this measure. Can we move to the next slide, please? Lastly, we looked at measures of mobility, and mobilization requires coordinated contraction and relaxation of multiple muscle groups. So it's an important measure of complex muscle function. We assess mobility using the 10-meter walk-run test and also the timed up and go, or TUG. In the 10-meter walk-run test, participants are timed over a 10-meter distance, of walking as quickly as they can, but comfortably without falling over. And in the timed up and go, participants start the test seated in a chair. They're asked to rise from the chair, walk 3 meters, turn around and walk back and sit down. And we time how long it takes to perform the test from beginning to end. So on the left is the 10-meter walk-run test. Here, the middle grey line is the placebo again. The bottom line is the 4 milligram per kilogram cohort, and the very top line is 2 milligrams per kilogram. Improvement is in a downward direction. This is actually the only test where we didn't see the 2 milligram per kilogram separating in the right direction from placebo. We're a little bit perplexed by that, but overall the data at 2 milligrams per kilogram is moving in the right direction. But I think if we look at the 4 milligram per kilogram, again, we can see clear separation from placebo at day 92, which is maintained through to 6 months. So maintained over that 3-month period of no further dosing at the end of the study. On the timed up and go, the colours are again the same. Improvement again is in a downward direction, and we can see that the placebo, or the grey line, doesn't really move very much at all, whereas moving downwards through the slide, the 2 milligram per kilogram and the 4 milligram per kilogram separate from placebo in the direction of improvement. At 4 milligrams per kilogram, this improvement is maintained over that 3-month interval where there's no further dosing, whereas at 2 milligrams per kilogram, the line starts to drift back to baseline. So what we've concluded from this, where we're seeing the 4 milligram per kilogram doses are maintaining treatment effect over that 3-month interval, but where we're seeing some loss of effect with the 2 milligrams per kilogram is that we really need to dose 2 milligrams per kilogram more frequently if that's the dose that we decide to move forward into our next clinical trial. And it's probably in the order of every 6 weeks for that dose, whereas we could comfortably dose 4 milligrams per kilogram every 3 months. Could we move to the next slide, please? So this is the last slide of my section. I'd first like to reiterate our gratitude to the participants and their families, the advocacy partners, as well as the study investigators and their teams for all of the hard work and persistence in the clinical trial. The MARINA open label extension study continues to follow the MARINA participants to evaluate safety and treatment effect over the long term. FDA has now eased the partial clinical hold that was placed on the program, and we're now able to dose escalate 12 participants to 2 to 4 milligrams per kilogram, which approximately doubles the number of participants that will have treated at the 4 milligram per kilogram dose in this study. We're actually well on the way with this dose escalation process, and the plan is to go back to FDA with that data as soon as we've compiled it. Additionally, we're quite a long way along now in our planning for a phase 3 study, which will most likely be the next study that we initiate with AOC 1001. We believe that the MARINA data that we shared today provides an excellent foundation upon which to base our phase 3 clinical trial design. We also continue to learn from our patient partners, and always looking for new ways to partner with the DM1 community, especially as we now move forwards to entering into phase 3. So finally, this is an exciting time for us at Avidity, and a really exciting opportunity for the DM1 community to collectively use our voice to raise awareness and work together to advance treatments for DM1. So thank you for your attention, and I'll now hand you over to Kristi, our Head of Clinical Operations.
Thank you, Steve. I'm so grateful to be here today to tell you about our approach to drug development and how you can get involved. Next slide, please. So this is a pretty standard line for drug development, but I want to tell you that 30 years ago, clinical trials were very different. You didn't really interact with the patients. It was sort of a taboo type of a thing. You really just conducted your clinical research and kind of hoped you were hitting the right marks. And now it's so different, and I think it's for the best. Patients are very involved. They're part of advocacy groups like MDF, and they're telling their story to the investigators and to the drug developers so that we can know what is critical to you in clinical trials and the treatment of DM1. So we spend a lot of time engaging with our patients after drug discovery, and we look for an unmet need such as DM1 where there are no treatments. Then we begin in the preclinical phase, really reaching out to foundations like MDF and talking with patients, finding out what's important to them. And not just the physicians, because they have a different perspective. It's your perspective that matters. You are the experts. So then we move on to the clinical trial process. And again, we're still engaging with our participants. And I want to tell you a little bit about some of the initiatives that we're undertaking as we move forward. Next slide, please. So we actively seek input from patient and caregivers. And on the right, you'll see a picture of our council for all 3 different programs that are currently in the clinic right now. And we look for feedback on protocol design. When I started here at Avidity in February 2021, I sat in and was able to listen to patient interviews and hear what their life was like living with DM1 and how the progression of their disease was affecting them and their family members. It was so important for the design of protocol. Also, we look at communications and patient-facing materials. We ask for your feedback, and that's been very helpful. We also offer, disease-specific focus groups and a rare disease muscle council, and we ask their advice on our trials and the questions that we're asking and our clinical endpoints. And we also ask patients to speak at our company-wide events, and this has been really, really fruitful. It really brings it home because every single day we focus on you, and that's really important to the fabric of Avidity. And we will continue to put our patients at the center of everything we do. In fact, in a couple of investigator meetings that are coming up, we have patients coming to speak to our vendors, and it really brings these studies to heart and to life. Next slide, please. As Lee mentioned earlier, we are very committed to our trial participants. So we have a travel concierge service because we've listened and we don't want you to have to pay out of pocket for expenses. So we also try to, if you do have to pay for out-of-pocket expenses, get you reimbursed quickly. We offer infusion day care packs, and they're age appropriate, so that you might have a blanket or some other comforts for that infusion day. And we're trying to reduce the travel to the clinic visits. So we have home health care, telemedicine visits, local lab draws, And trial visits with local primary care physician is something that we're working on for the future. So instead of traveling to a KOL or a center of excellence, you might be able to go to your primary care physician for some simple blood draws. So those are some things that we're working on for the future. Next slide. So also in our commitment to trial participants is that we understand that we want to improve the clinical trial experience. So we've heard from our patients and folks living with DM1 that you want to get your trial results. You want to get them personal. You want to know, were you on placebo or were you on active? You want to get your genetic testing results, and you want to hear what your individual assessments were like. How'd you do before participation or enrollment in the study, and how you did by the end. And you want to hear from the sponsors, and that's really important. And the other thing that we recently learned is it's very important to have a personal connection with your investigator or your physician that's enrolling you in the trial. So in order to engage patients during a clinical trial, which can be really troublesome, to be honest with you, because we can't have direct contact with patients, we've actually engaged with a vendor that can do surveys during the clinical trial to see how things are going from your perspective. It also gives us an opportunity to thank our participants, because recently we wanted to be able to do that on the MARINA trial when it was closing, and we couldn't do that without getting an approval to do so from an IRB, which is an institutional review board that protects patient safety. And so we wanted that way to connect with our patients. So that's something that we'll be implementing in our future trials. And this helps us make each trial that much better. Next slide. So there's some ways that you can contribute to the development of new therapies. So one is that you can connect with your medical facilities, experts in DM1, and learn about ongoing research. It's super important. Also, one thing that's critical to the success of our programs is that you participate in data gathering, surveys, interviews, focus groups, and observational trials, because we take that data and we comb through it. Our biometrics group is looking through that data to understand the disease progression and to look at clinical endpoints that could convince the regulators that our therapy is making a difference in your day-to-day lives. Also engage with groups like MDF and participate in activities to support, learn, and share with each other. And so I'm sure when there are new families that are diagnosed with DM1, there are many of you that reach out and help and offer support and tell them where to start looking for help and treatment. And then also mobilize within your community to advocate. So kind of like MDF is going to the Hill, like participate in those types of things to let the regulators know that you're really looking for a treatment, and here's how we're going to do that. Very important. And share your knowledge with others. I mean, you are the experts in your disease, and it is so crucial that you share that. So that's all I had to say. And I just want to tell you how grateful I am to all of you living with DM1 and all of the patients that participate in the focus groups, the natural history studies, and our clinical trials. Thank you.
Thank you, Kristi. We want to thank each patient and their families for the willingness to contribute to the DM1 community and the work that the community does through participating in surveys, natural history, studies like NDM1 that are so important, and supporting patient advocacy groups and the incredible work they do, such as the Myotonic Dystrophy Foundation. All of these activities are really indispensable when it comes to finding therapies for DM1. If we can go to the next slide, please. So I wanted to close out with just, this is us at Avidity. This is your team and your partners that are committed to working with you with the goal of ultimately being able to improve the lives of families living with myotonic dystrophy. At its simplest form, we are people looking to try and help other people. And the way we're doing that is through working on our science, advancing our technology, delivering therapeutics into the clinic, and then also our commitment to develop those therapeutics the right way, but also as quickly as possible, as we know there's a whole community waiting. And with that, I'm going to hand over to Luis as we move into our Q&A session.
Thank you, Sarah. Hi, I just wanted to introduce myself to everyone. My name is Luis. I'm a member of the patient advocacy team at Avidity, and I just want to extend a thank you to everyone for joining this webinar today and for submitting your questions ahead of time. Just as a reminder, you're welcome to continue submitting questions using the questions tab on the control panel on your screen. Feel free to type it in and then type your question and click send. And now at this moment, I'd like to invite all of our speakers to just come back on so I can start directing questions your way. So thank you very much. I'll start off with a question just for Sarah. When is phase 3 clinical trial scheduled to begin, and how can one enroll?
That's a great question. And one of the elements I think as Steve spoke to were deepen that process of looking at phase 3 design and what the study will look like and the planning around that. We'll need to go through a set of discussions with regulators around agreeing that design. As soon as we have updates and information, we are committed to providing those in real time and as soon as possible to the community. And please rest assured that when we're ready to be up and going through the study, that information will be available around recruiting sites and also how to get involved. I just want to convey that we are working as expeditiously as possible. We understand the urgency and we're deep in that planning process right now and working really hard towards getting going.
Thank you, Sarah. The next question will be for Steve. Will the treatment reverse, or do we anticipate the treatment reversing symptoms from patients?
Thanks for the question, Luis. That's a difficult one to answer. The MARINA study, in terms of studies for neuromuscular type diseases, was relatively short to really, and it wasn't really possible for us to evaluate the full treatment effect as it might happen over time. And we would anticipate that the effects we saw early in the MARINA study may well continue to improve as we go out longer in follow-up. So we'll have more of a clear idea of that as we can move through phase 3. But certainly we did see movements within the MARINA study that suggested improvement rather than stabilization. We hope it's possible to see improvements in the disease as we go out over time in a longer, larger study.
Thank you, Steve. And then I have another question for you from the audience, and that is, currently patients are receiving 2 mg and 4 mg per kg doses. Do you foresee these dosages increasing or changing as we move into phase 3?
That's a good question. We finished our dose exploration. So we finished our dose ranging within the MARINA study, and we've concluded that the dose that we would select for a phase 3 study would be somewhere in the 2 to 4 milligrams per kilogram range. And as I mentioned, if it's 2 milligrams per kilogram, we'd need to dose a little bit more frequently than we would dose with 4 milligrams per kilogram. We don't anticipate the need to go higher than 4 milligrams per kilogram. As I said, we've finished the dose ranging exploration, and the dose that we move forward into phase 3 with is something that we really need to discuss and agree with FDA along with the overall design of the clinical trial.
All right, thank you for that. This question I can direct to Lee. Can you speak to what went into the inclusion criteria for MARINA and what goes into inclusion and exclusion criteria moving forward?
So thank you for the question. This is a very good question because the inclusion-exclusion criteria for a clinical trial is likely to affect the outcome of the trial. So we spend a lot of time thinking about these criteria. And we really leverage several sources to understand how to determine these criteria. First, we use the natural history study data that's available to us to understand where the patient fits and how many patients in what category and how likely they are, what's their disease status over time. We also talk to clinicians who see a lot of patients and have a lot of experience with DM1 to understand the anticipated progression in their clinic. And lastly, we talk to patients to understand what's the impact of certain inclusion-exclusion criteria and how this might affect their life or their participation. So this is what we did with the MARINA trial. Prior to trial start, we had really many days, many months of discussion and investigation to get to the criteria that we determined. We're in the process of doing the same with the next trials that we're planning. So, and there are a lot more consideration because the trial will be likely to be a little bit larger. So, so those things will, become available, and as soon as they're available, we will share with the community so that you can have a sense of where how this trial could benefit patients and people you know. Thank you.
Thank you, Lee. We have time for a few more questions, and this one I can direct to Sarah. Do we have plans or have an idea of where we want to move forward with phase 3?
So in relation to work, it's likely to be a study that will have many sites, both in the US and then also outside of the US. So we're looking to build a global study that involves many countries and investigational sites. And Kristi and her team are working really hard on that. I see that big smile from Kristi. So yeah, we're looking forward to having something that is, as Lee also said, likely to be larger than the MARINA study as well. So that's both in terms of patient numbers as well as the number of sites and the number of countries.
All right, thank you so much, Sarah. I have one more question I can direct to Steve. Is there evidence of AOC interacting with healthy or normal RNA?
No, the AOC 1001 interacts with both the wild type, which is the normal copy of DMPK RNA, and also the mutant copy of DMPK RNA. We don't have an ability to discriminate between the two. And that's similar for all of the antisense approaches currently that are either in the clinic or approaching the clinic. It doesn't interfere with other RNAs, and we will have thousands and thousands of RNAs as are making every single day. And so AOC doesn't interact with any of those others, and we screen for that as part of our screening process before bringing drugs into the clinic. In terms of the consequences of knocking down the wild type DMPK messenger RNA, that's been studied extensively in animals by both us and other people that either are or have developed drugs in the past for DMPK, for DM1, and there doesn't seem to be any adverse effects of knocking down the normal copy of the DMPK messenger RNA.
All right, thank you. And then we have time for one more question, and this one will be for Kristi. How can the community continue supporting Avidity and getting involved?
I think one of the best ways is if you decide you want to participate in a clinical trial, to really look at the scheduled events, the assessments, make sure it's right for you and your family, and realize that every assessment that you undertake, and if you have biopsies or whatever it is, just realize that that is your voice. So if you're filling out forms, take it to heart, because we look at every single piece of data, and every single patient matters so much. So that would be one piece of advice. The other is just to keep getting involved, especially those natural history, because we do look at that data. It's very important. And thank you.
All righty. Well, thank you so much, everyone, for answering the questions. I think that's the time that we have today. So we'd like to welcome Tanya back.
Thank you, Louise. Thank you all. This has been fantastic. We appreciate you sharing more about MARINA Trial, MARINA OLE, and AOC 1001 overall. The results are very exciting, and we're looking forward to hearing more in the near future, and looking forward to seeing you all in Washington at our conference next month. Thank you to our speakers, to everyone who's joining us today. I hope to see you all in Washington, D.C. next month, and I would love to wish you all a fantastic weekend. Thank you so much. Take care.